Our mission at Aletira Therapeutics is to use alternative splicing to advance life-changing gene therapies. To understand our value proposition, it's helpful to understand how gene therapies are already having an impact. Adeno-associated virus or AAV-based gene therapies are now preventing some forms of previously untreatable vision loss. And children with a rare form of congenital deafness can now hear for the very first time. Despite this tremendous progress, we still do not have effective gene therapies for many diseases, including major forms of hearing loss, vision loss, and many others. There's also untapped potential of gene therapies for many other applications. One of the major challenges that has constrained the reach of gene therapies is limited cell type selectivity of existing platform technologies. For example, most advanced gene therapies use AAVs that comprise a capsid and a DNA payload consisting of a promoter that drives gene expression. These have been highly successful for some applications where you simply need to replace a gene. However, for other applications where you need a gene in a target cell that is toxic to other cells, they often fall short. And the limitation has to do with the cell type selectivity that doesn't prevent them from getting into those other cell types. Now, there have been advances in capsid technologies that can essentially get the adenovirus, AAV, into the right tissue, essentially the right zip code, but essentially not the right street address, that individual cell type. And there have been advances in promoter-based strategies where that can be optimized for selectivity. However, this often comes at the expense of gene expression, a major compromise. The other main challenge is that with these existing approaches, development is often slow due to the empirical process, and these don't necessarily translate well across species. So to address these challenges, we're using a complementary approach that leverages alternative splicing, which is the natural mechanism by which different cell types use different exons to create unique protein products. The approach pioneered by our scientific founder, Professor Geoff Lynn, is to take target cell-selective exons, referred to as selexons, and insert these between a promoter and transgene. The AAV can go into multiple different cells, but the gene is only expressed in the target cells that use that selexon. This offers improved selectivity because splicing is tightly regulated at the cell subset level. We also don't have a trade-off in potency because we can use strong promoters together with the selexon. And then importantly, because splicing is relatively conserved across different species, we don't have an issue with translation. We've now developed a computational platform referred to as Selexer that allows for the rapid identification and optimization of these selexons for different applications. Our general approach is to query curated databases to identify target cell type specific exons, and then use machine learning to trim these into novel and proprietary compositions of selexons that can be incorporated into gene therapy platforms. With this approach, we've already identified about 400 unique selexons covering different tissues and cell types, and this number continues growing as we continue to integrate both publicly available as well as internally generated data. Indeed, this approach allowed us to select our lead candidate targeting GJB2 mutations, which accounts for a major form of hereditary deafness, and is also an excellent use case for this technology. For example, in normal hearing, sound enters the external ear and is detected as vibrations in the inner ear by hair cells that transmit the signal to the CNS. In patients with GJB2 deficiency, the supporting cells, abbreviated SCs, that surround the hair cells are not able to perform vital functions such as ion regulation, and this eventually leads to progressive hearing loss. Now, it's simple enough to just take an AAV and reintroduce GJB2 into supporting cells. The challenge is that GJB2 is toxic to the critical and neighboring hair cells. So for this application, selectivity is key. Fortunately, we've developed a construct referred to as SLX01 that uses a selexon for supporting cell-specific expression. A version of this construct expressing green fluorescent protein was shown to have high supporting cell expression shown here with undetectable expression in neighboring hair cells. Importantly, we also see conserved and high levels of expression across multiple supporting cell subsets, whereas a competing promoter-based strategy shows variable levels of expression. We've also now shown that this construct expressing GJB2 has similar patterns of expression and expect efficacy data early next year that would trigger development studies that would allow us to be on track for IND submission in 2027. Now, in terms of market potential, GJB2 is the most common form of congenital deafness, accounting for about 3,500 cases per year in the US. And the standard of care cochlear implants does not restore natural hearing, so we would expect with a potentially curative therapy, there would be high rates of adoption, with a market exceeding at least $500 million per year. As evidence of this market potential, two of the most advanced companies developing otoferlin or OTOF-related deafness therapies, which is about 1/10 the market size of GJB2, were each acquired for over $200 million at the preclinical and early phase 1 clinical stages. Additionally, Skylark Bio raised a $40 million Series A just last year to develop a promoter-based strategy targeting GJB2. In terms of competition, there's at least 4 other companies, but all of these companies are at the preclinical stage and using promoter or capsid-based strategies that have the limitations discussed. Selexons offer a differentiated approach that allows us to be competitive in a very compelling opportunity. And the other point I want to emphasize is that this is really just the beginning for this technology. We've already demonstrated that Selexons can be used for targeting photoreceptors for vision loss applications. We can target specific neuronal cell populations for CNS applications, and we can also improve the selectivity of oncolytic viruses. All of this is described in an original publication by Ling et al. In Nature Communications in 2022. As part of our ongoing R&D efforts, we aim to unlock their full potential, using Selexons initially for priority indications for hearing and vision restoration, but we also have moonshot indications going after, for example, senescent cells to target aging, to try to prevent neurodegeneration, as well as looking at oncology applications. Another point I want to stress is that we are initially using AAV, but this is not an AAV-restricted technology. We aim to evaluate this technology for other gene therapy applications, including gene editing, mRNA, as well as other platforms. Now, this may seem ambitious for a small company, but Selexor allows for rapid identifications of Selexons that can be tested in vitro and that translate well across species. This will allow us to create a funnel for a compelling pipeline of internal candidates, but most importantly, Selexons are complementary to other gene therapy technologies. So we do seek to use this technology as a platform to seek partnering opportunities for non-dilutive financing as well. To help fund these efforts, we expect to soon close on a pre-seed round. We were founded about 2 weeks ago, incorporated, and expect to soon close on this round that will allow us to de-risk our lead SLX01 and expand our platform and strengthen our IP portfolio. That will put us in a strong position next year to raise a larger seed that will allow us to bring in a development and management team, as well as to de-risk our lead up to IND enablement and select a lead for vision loss. That would create a compelling pipeline that we believe would put us in a strong position for Series A financing to move our lead from phase 1/2A to 2B and advance at least one additional candidate to clinical testing. With emerging clinical data, we think that would be a catalyst for partnering opportunities, but it also might trigger acquisition interest as well as the ability to go public, hopefully in better market conditions. Fortunately, we've really had tremendous support from many of the folks in this room, including Blackbird and MII, that have provided catalytic support in the form of funding as well as a dedicated team, including many members of the audience, especially Matt Lawler, who found this project and helped develop it with Professor Ling. And that's really creating strong foundations for us. Importantly, technology is clearly differentiated with also a very compelling value proposition. And we aim to protect that with a layered IP strategy consisting of foundational IP out of Hopkins, as well as more specific patents going after specific use cases and compositions. In terms of the environment, fortunately, we will benefit from continuity. We have ongoing collaborations with Hopkins that allow us to continue moving our R&D programs forward as we build out our space at Blackbird's incubator site that's set to open early next year. To help kind of realize this ambitious goals that we have for the organization, we're relying on initially strong support from our scientific founder, Professor Ling, who's been the force behind this technology for many years now. We also have Virginia Berger joining as the founding Chief Technology Officer. Virginia developed Selexor and brings industry experience, having led New Equilibrium as CEO previously. As for myself, as founding CEO, I'm bringing development experience, having built platform technologies from early stages of discovery all the way up to phase 2 clinical testing, and directly relevant. Just several years ago, I had founded the company Avidya Technologies here at Hopkins and led it from 2017 through to our exit in 2021. Avidya Technologies was a platform technology company with many parallels, and I'm hoping to draw upon that experience in building and ensuring the success of Aletira. This time, though, I'm excited to additionally have Hopkins support again, but also now having the support of Blackbird and hopefully have the support of many of you as well. So I'll stop there and welcome any questions you have, and also have some of the co-founders come up and join me on stage. Thank you.